Okay, so good evening, everyone. So I welcome you all to the week 10 problem solving session of this NPTEL course Molecular Biology. So I hope all of you have gone through the week 10 lectures. I will wait for 3 to 4 minutes and then I will start today's session. So I'll just wait for other learners to join. Okay, so I will share today's slides. Okay, so good evening, everyone. So today we are in the week 10 problem solving session of this NPTEL course Molecular Biology. I'm Akash Jyoti. I'm one of the PMRFTAs for this NPTEL course. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. Let's first have a quick recap of what all we learned in week 10. So in week 10, we were covering the topics that were covered were was mainly molecular cloning. So what are the various steps that are involved in molecular cloning? So once you, one second. So can anyone just say, summarize what all have been covered in week 10? Okay. Basically, this is basic biotechnology you have already in class 12 as well. So in molecular cloning, what is done is basically once you have your gene, once you identify your gene of interest, or you have identified the particular gene corresponding to your protein of interest, then what is done is this gene is cloned into a vector, right? You clone the gene into a vector. Okay, the gene is cloned into a vector, and then what is done? So once this gene is cloned in the vector, the cloned vector is then transformed into a host cell. Okay. This is transformed into a host cell, and then once it is transformed in the host cell, then the protein can be expressed. You express the protein in an expression host. Okay. So this is the overall um, summary of what is done in cloning. Okay. So now this vector can be there can be different type of vector. There can be there can be a bacterial vector like plasmid. Plasmid is one of the most commonly used vectors. Then there can be mammalian vector. There can be other eukaryotic vectors can also be present. So depending on what is your protein, or what is the protein you want to express, or what is the size of your gene, so all these factors will regulate what type of vector you will choose. Okay. And then the host cell is also, there can be different types of host cell. It can be a bacterial cell, it can be a yeast cell, it can be a mammalian cell. So if you are cloning it in a mammalian cell, you will need a mammalian vector. Okay, so you cannot use a plasmid vector for you cannot clone your gene in a plasmid vector and then transform it in a mammalian cell. Okay, so there are different types of mammalian cell vectors that you have to use if you want to transform your clone gene in a host cell, in a mammalian cell. Okay, so it depends on what is the type of protein you want to express, what is the size of your gene. So all these factors have to control the uh, the suitability of the vector, and then the what type of host cell has to be chosen. Okay, and protein overexpression, this is done by using IPTG, isopropyl thiobeta galactopyranoside. Okay, so when this is analog, this is an analog of allolactose. Okay.
So the host cell and expression host cell are different.
Yes. Yeah. Host cell. Actually, there are in if you look in case of bacteria, so there are two types of host cell. One is one is your expression host cell. Okay. And another is just for storing your plasmid. Okay, storing the vector. So if you when you go to work in a lab, so you will find that there are I'm saying in case of E. coli cell. So in case of E. coli cell, there are different types of strains, engineered strains that are used in a laboratory. So there are strains like BL21DE3. Okay, BL21DE3. Then there are RIPL cells. Then there are RIL cells. These are the names of different strains of E. coli. So these are the expression host cells. And then there are host cells where the plasmid is only stored. Like there are DH5 alpha cells. Okay, so these host cells. So if you transform your plasmid in these type of host cells in DH5 alpha cells, then your protein will not be expressed. So if you want to express your protein, then you have to transform your plasmid. You have to transform your plasmid in these types of host cell, BL21, RIPL, RIL. So there are different types of so expression host cell is different from uh, the host cell where you just want to maintain the plasmid. So for example, you have created a clone. Okay, you have cloned your gene of interest in a plasmid and you want to store that plasmid for a longer duration of time. Then what is done is this plasmid is transformed. It is transformed, or it is this introduced in DH5 alpha strain of E. coli. Okay, so these are engineered cells and they have certain mutations which prevent the plasmid from getting degraded. Okay, they are maintained. They can be maintained in these types of cells. Okay, is it clear? So these host cells, these are commercially available actually, BL21, RIPL, um, then RIL. So these are, so the details, I think they will be covered in later lectures.
So, sir, means in the
Yes.
case of prokaryotes only, or in the case of eukaryotes also, the vector and host cells are different.
In eukaryotes also, there are, like in mammalian, not very sure about mammalian cells. Uh, so in case of mammalian cells, I have not worked much with mammalian cells, so I'm not very sure about whether there are like host cells. There are two different types of host cells in case of mammalian cells, but in case of bacterial cells, prokaryotes, there are two different types of strains that are used. Okay, but
Mm.
mm,
So, sir, DH5 alpha is bacteria only.
yeah, DH5 alpha is E. coli cell only. These are all E. coli cells, but these are engineered. Okay, these are engineered E. coli cells. So there are certain mutations have been done so that they can overexpress the protein, or so that the integrity of the plasmid can be maintained. So these are all E. coli cells only. DH5 alpha. They are the names of different strains. Okay.
Okay, sir.
Okay. So what is transformation? You all know, right? So just can you give a brief? Can you tell briefly what is transformation? Because we have already covered these topics earlier also. What is
So, uptake
transformation?
of
Yes.
DNA from the surroundings.
Yes, yeah.
If that
Transformation is,
is
hmm. So bacterial cells have this property that they can uptake the DNA which is present in the surrounding environment. Okay, but the problem is that there are only very few naturally. So naturally, yeah, there are certain bacterial strains which can take up DNA very easily, but there are very few. But if you want to transform any plasmid into a bacteria, so just you have to chemically treat the bacteria. Okay, the bacteria will not take any plasmid from the surrounding. It is difficult difficult to transform any plasmid in bacteria. So you have to chemically treat the bacteria so that the bacteria can uptake the plasmid from the environment. environment. Okay, or DNA from the surrounding environment. So most species of bacteria, including E. coli, take up only limited amounts of DNA under normal circumstances. So to transform these species efficiently, the bacteria must undergo some physical or chemical treatment that enhances their ability to take up DNA. So you have to chemically treat the bacteria with certain type of chemicals and reagents. So these chemicals and reagents make the or increase the ability of the bacteria to uptake DNA from the environment. So what is done is usually for in case of bacterial cells, there are different. So it depends again. It depends on the host cell. So if it is bacterial cell, then if you treat the bacterial cell with calcium chloride, or there are different divalent salts like rubidium chloride, calcium chloride, all these salts are used for so if you these type of chemicals are used only in case of bacterial cells. When the bacteria is treated with these type of chemicals, what it does it this cell wall, the cell wall that is present in the bacteria, that is somewhat it gets loosened. Okay, so what happens? So salt treatment induces an overproduction of certain outer membrane proteins. So the correct reason as to how calcium chloride treating the calcium chloride uh, 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 how treatment of calcium chloride increases the efficiency of bacteria to take up plasmid, that is not clear. But what is thought is that this calcium chloride may induce an overproduction of certain outer membrane proteins. So once there is an overproduction of outer membrane proteins, then what happens? This DNA is added to the treated cells. It remains attached to the cell exterior and is not. And at this stage, it is not transported into the cytoplasm. So the DNA. So once you treat the bacteria with calcium chloride, then what happens? It might happen that there is some expression of outer membrane proteins, and also after this treatment, the DNA which is present in the environment, they gets attached uh, to the outer wall of the bacteria, but they are not uptaken by the bacteria. So for uptaking the plasmid, for uptaking the plasmid inside the cell, what is done? A heat shock method is a heat shock method is followed. So what happens? What is done is so what is so what so first you have the bacteria. Okay, you have a culture of bacteria. You treat it with calcium chloride. Okay, once you treat it with calcium chloride, the bacterial cell are now called competent cells. Okay, why is that called competent cells? So they are called competent cells because on treating the bacteria with calcium chloride, the the ability of the bacteria to take up the DNA from the outside increases. So now they are called as competent cells. Now what you have to do, you have to take these competent cells and then you have to follow a heat shock method. So what is done is that these competent cells are first placed in ice for around 15 minutes. And then after placing it in ice for 15 minutes, then the plasmid is added to the bacteria. After adding plasmid to the bacteria, then the bacteria is given a heat shock. So the bacteria is placed in 42 degrees Celsius water bath for around 90 seconds. And then after the 90 seconds is over, you take out these competences, you take out this bacteria and then place it in ice for another 20 minutes. So you give a heat and then you again place it in ice. So this method it facilitates the DNA to be taken in by the bacteria. Okay, so the actual movement of the DNA into competent cell is stimulated by briefly raising the temperature to 42 degrees Celsius. So it is possible that this heat shock changes the permeability of the membrane to DNA. So when you apply this, give this heat shock, the permeability of the membrane to DNA it changes, and as a result, it induces the activity of membrane protein to transport the DNA into the cell. Okay, so there are different reasons. So some say that the DNA is negatively charged and calcium is positively charged, so it facilitates the uptake of DNA into the cell. And then there are some school of thoughts which say that this calcium chloride treatment induces the expression of some outer membrane proteins, which facilitates the DNA to be uptaken by the cell. So, so this is the, but what happens is this is the method that is followed, and this method, by through this method, you can transform or you can inject or you can transfer your plasmid into the cell. Okay. So first you treat it with calcium chloride. So on treating with calcium chloride, the bacterial cells become competent. Competent means that they are now competent to accept the DNA from the environment. And after that, you give a heat shock. So first you place the bacteria, you add plasmid into the bacterial cell, competent bacterial cell, and then you give it a heat shock for at 42 degrees Celsius for 90 seconds, and then again you place it in ice. So repeated heat and shower. Then first you heat it and then you cool it, and this causes the cell wall to become loose, and the DNA can enter inside the cytoplasm. Okay. Now after so how will you now there has to be some now when you are doing this transformation
Right.
there will be
The
yes.
in water bath we do this, no? Means first.
Yes, this 42 degrees Celsius heat shock. So there is a water bath, and this the temperature of the water bath is set to 42 degrees Celsius. And in this water bath, you place this microcentrifuge tube for 90 seconds. Okay, for around 90 seconds, and then you take out this microcentrifuge vial and then again place it on ice. Okay, for another 15 minutes or minimum you have to keep it for 5 minutes. Okay.
So before the water bath means before doing
Hmm.
42 degrees Celsius, before
Hmm.
giving, uh, we inoculate the plasmids in the means
Yes.
in the test tube itself.
In
In
the
the sorry
competent cells.
centrifuge tube.
Yeah, in the mic, this MCTs, these Eppendorf tubes, if you have ever seen. So, in this, so you take the plasmids, around 100 nanogram of plasmid is 100 to 200 nanogram of plasmid is required for transformation. So you take this much amount of plasmid and then you add it into the competent cells, the competent cells that you have prepared, E. coli competent cells. You add this plasmid.
So nothing happens to the plasmid means when
So,
we means before is in the starting we
so the plasmid. So you have this bacterial culture, right? And when you add this plasmid, then this plasmid will remain in the environment of the bacteria. Now you have to do a process so that this bacteria can uptake the plasmid from the outside into the cell.
also outside means means plasmid will remain viable for like we are
Yeah,
taking the
plasmid
plasmid,
is
it's only the plasmid, so will it be viable?
yeah. Yeah, it will be viable. No, it is DNA. What is plasmid? Plasmid is circular double-stranded DNA.
Hmm.
And DNA remains stable at very high temperature. So at uh, at 42 degrees Celsius, it will not get denatured. Nothing will happen.
So are we using some preservatives or buffer to keep the plasmid?
Mm, no, no, no. So there are different kits. If you have worked in any laboratory, so what is done is there is a specific protocol for isolation of plasmid. So there are kits available actually. There are mini prep kits by which you can isolate the plasmid from bacterial cells. So once you isolate this plasmid is stored in -20 degrees Celsius. So if you want to, these plasmid remain stable even at 4 degrees Celsius. But if you want to store it for longer periods of time, then this isolated plasmid is stored in these microcentrifuge tubes at -20 degrees Celsius. Okay. So you directly take out this plasmid and you add that particular amount of plasmid into the uh, competent cells. And then you give heat shock to the competent cells and the plasmid will be uptaken. Okay. So
Okay.
nothing will happen to the plasmid. They are double-stranded circular DNA, no? So they remain stable. Yes. Any is your doubt clear?
Yes, sir.
Okay. You do not need. And the competent cells, so what is done is the competent cells are they are a glycerol stock is made. So if you so once you have given treated this E. coli cells with calcium chloride and you want these competent cells, you want to store the competent cells. So for storing the competent cells, glycerol stocks are prepared. So these competence cells are stored in 20% glycerol stock. Okay. So you take the so if you are preparing like 15 ml of competent cells, then accordingly 20% of 15 you can accordingly you can do the calculation and then prepare glycerol stocks of that. So the competent cells has to be stored in glycerol stocks, 15 to 20% or 20 to 25% glycerol stocks, but the plasmid you can store it as it is. Okay. So now, when you are doing this transformation experiment, there not all the bacteria will take up the plasmid. Okay, so there will be only a very few bacteria which will take up the plasmid from the environment. Uh, so, uh, uh, so, so what is done is after transformation. After transformation, the culture that you have, you plate it in an agar plate. Okay, so there are agar plates, and these agar plates are supplemented with some antibiotics. Okay, so for example, the plasmid. So for example, you have cloned your gene of interest in a. So this is your gene of interest. Okay, this is your gene of interest, and this is your vector, plasmid vector. Okay, so for example, this plasmid vector has a particular antibiotic resistant gene. Say it has ampicillin resistant gene. Okay, so uh, and now you have transformed this plasmid into the bacterial cell. So you have a bacterial cell, and this plasmid has entered into the blast into the E. coli cell. Okay, so this is your E. coli cell, and this plasmid has entered this E. coli cell. Okay. Now there will be other bacteria also which will not have any plasmid. Empty E. coli cells without any uh, yeah, without any plasmid. Okay. So what you have to do is you want to isolate. You want particularly these type of bacteria. These are called transformants. Okay. So transformants are the bacteria taken your which have taken the plasmid, which have taken up your recombinant plasmid. Okay, and these are non-transformants. Non-transformant are those bacteria which have not taken up the plasmid. Okay, so you want to specifically select these bacteria, and you do not want these bacteria. Okay, so what you will do, because this bacteria has now ampicillin resistant gene. So if you plate this bacteria in agar plates containing ampicillin media, then only these type of bacteria will grow. Okay, so you will get colonies. You will get colonies for only these type of bacteria. While these bacteria, they will not survive in ampicillin supplemented agar plates. So in this way, you can screen or you can specifically isolate these bacteria and remove these type of bacteria which have not taken up your of the cloned gene of interest. Okay, is it clear? So So here, this is actually, these figures have been taken from the book T.A. Brown. So here they have given an example of PBR 322. So if you see the PBR 322 is a plasmid vector. Okay. So PBR 322 is a plasmid vector, and this plasmid vector has two type of antibiotic resistant gene. Okay. It has an ampicillin resistant gene and it has a tetracycline resistant gene. Okay. So after a transformation, so if for example you are using this vector, PBR 322 vector, so only those E. coli cells that have taken up a plasmid, only they will be ampicillin resistant and tetracycline resistant. So if you use this vector and then you have transformed it in an E. coli cell, and then you grow the E. coli cell in an agar plate having ampicillin and tetracycline, then what will happen? Only those bacteria cells which have taken up this PBR322 vector, only they will grow. Okay. Non-transformants are still ampicillin sensitive and tetracycline sensitive, so they will not produce any colonies. So in this way, transformants and non-transformants can be distinguished from one another. So this figure, you can see this figure. So this is a normal E. coli cell which has not taken up any plasmid. Well, this is an E. coli cell which has taken up this PBR322 plasmid. So this E. coli cell is now resistant to ampicillin and tetracycline. So when you plate it on an agar plate, only these colonies will be these colonies will be present, and these colonies will not be able to survive. They will die, and you will not observe colonies of these type of bacterial cell. Okay, is it clear?
Okay, sir.
Okay, these things are easy, no?
Yeah.
Okay, so now what is recombinant screening then? So what happens is when you are doing any cloning, when you are doing any cloning, sometimes what happens? So if this is your vector, okay, this is your vector, and this is your gene of interest. So this is your gene of interest. Now what you do is you treat your vector. You treat your vector with the same restriction enzyme that you treat it with the restriction enzyme that you treat with your gene of interest. Now then you do ligation. You set up a ligation reaction. So once first you treat it with the restriction enzyme. So once you treat it with the restriction enzyme, you will get a this circular DNA will open up, right? And also similarly here also you will get some sticky ends. Okay. Now when you do a ligation reaction, what will happen? These two will you expect the vector to ligate with your gene of interest. The vector gene should ligate with your gene of interest. Okay. And this is your recombinant plasmid. So this is your recombinant plasmid. But there will be, but when you're doing cloning, not all the plasmids will be recombinant. There will be some plasmid which have self-ligated. Self-ligated means they have which means the gene of interest has not ligated into the vector. The vector has ligated on its own. Okay, has ligated on its own. It does not carry the gene of interest. So how will you isolate these type of vectors from these type of from the so how will you differentiate the recombinant plasmid from the self-ligated plasmid? So for that, what is done is so for that what is done is basically in a lab what we do or what normally what is done is you will have different type of colonies. Okay, you will get around 5 to 6 colonies. Now you isolate, you pick up these colonies, and after you pick up these colonies, then you culture it, you inoculate it in LB media. Okay, you inoculate in LB media. Then once the culture grows, what is done? You isolate the plasmid. You isolate the plasmid from this culture. Okay. Now using this plasmid, what you can do? You can do a restriction digestion. Okay. You can do a restriction digestion. So what will happen if you do a restriction digestion? So if it is a recombinant plasmid, if it is a recombinant plasmid, then what will happen? You will observe two bands when you run an agarose gel. You will get two bands. One is the backbone band, and another is the insert. Okay, but if it does not, if it is a self-ligated plasmid, then there will be a single band. Okay, there will be a single band that is corresponding to the vector, that is corresponding to the vector size. Okay, but if your plasmid has if it is a recombinant plasmid, then one band will be of the insert, and another band will be of the backbone fragment. Okay. Or another method that is done is PCR. So you take a reverse primer and forward primer, and then you set up a PCR reaction. So if your plasmid has the gene of interest, then on PCR amplification on running the PCR amplified product through the gel, we will observe that particular DNA size bands. Okay, so if your gene of interest is say uh, 1 kb, okay, then you will get 1 kb amplified products, 1 kb amplified bands on running an agarose gel. Is it clear? So let's see this figure. So this is the vector molecules. These are the vector molecules. So you have treated the vector molecules with the restriction enzymes. Now, on treating this vector molecules with restriction enzymes, they have opened up. Okay, they are unligated. Now you add your gene of interest. Now, when you add your gene of interest, there will be three different population in your sample. There will be one population which will have self-ligated vector molecules. So these will not have your gene of interest. Okay, then there will be one population where you will get your desired plasmid, which is the plasmid having the gene of interest cloned in it. And then there will be wrong recombinant DNA molecules. So the gene of interest has not got cloned other gene of interest, or it has get uh, the wrong gene of interest has been cloned in this plasmid. So these are the type of or it has entered in the wrong site. So there can be wrong recombinant DNA molecules will be also. So now all these circular molecules will be cloned. Okay. Now because these vectors will have your resistant genes, so you will get you will get colonies of these population also, these population also, and these populations also. But what is desired is this one. You want only these type of colonies. You don't want these type of colonies. Okay. So cell containing self-ligated vector will also have give you a clone. You will colonies, and cells containing the desired molecule will also give you colonies, and cells containing wrong recombinant DNA molecule will also give you colonies. So
Uh,
there has yes, tell me.
So, what is the difference between the previous slide and this slide? The same thing only.
No, it's not the same thing. Let me explain you. And so there are two concepts that you have to understand. Okay, one is what is the meaning difference between transformants and what is the meaning of recombinants. So the meaning of transformants is you have an E. coli cell. Okay, and the E. coli cell, if the E. coli cell has taken up your plasmid, has taken up the plasmid, then it will be called as transformants. But there can be a situation where your where the plasmid where the plasmid does not have your gene of interest. Okay. Ligation. So there can be understand that there can be two different populations. In one population, the plasmid has your gene of interest. This is your gene of interest, and this is your vector. And there can be another population where only the plasmid has self-ligated. Self-ligated means your gene of interest has not ligated into your plasmid. The ligation. So when you set up a ligation reaction, it does not ensure that all the plasmids will be all the gene of interest will be ligated in the vector. There will be some plasmid where which will be a self-ligated plasmid, which will not have your gene of interest. Okay, and then there can be other plasmid where the gene of interest has entered in a wrong location, or any other gene has got inserted in this vector. Okay. So this is wrong gene of insert. This is not your desired gene of interest, or it has got inserted in a wrong location. So there can be these three different type of population. So now when you transform your bacteria, then there will be some bacteria which will have these type of plasmid. Okay, there will be some bacteria which will have these type of plasmid, and then there will be another type of bacteria which will have these type of plasmid. Okay, and all these three different bacteria will give you colonies. Why will it give you colonies? Because all these bacteria have these ampicillin resistant gene or this tetracycline resistant gene. वेक्टर जो है ना, वेक्टर में अगर एम्पिसिलिन रेजिस्टेंट जीन है मान लो, तो ये तीनों ही ग्रो करेगा ना आगार प्लेट में। तो आगार प्लेट में तुमने एम्पिसिलिन डाल दिया, तो ये वाले जो बैक्टीरिया है ना, ये वाले जो बैक्टीरिया है ये भी ग्रो करेगा, क्योंकि इसके पास भी एम्पिसिलिन रेजिस्टेंट जीन है, तो ये भी तुम्हारे आगार प्लेट में ग्रो करेगा जिसमें एम्पिसिलिन रेजिस्टेंट एम्पिसिलिन डाले
हाँ
हो
सर,
तुम।
हाँ सर,
ये
वो
भी
समझ आ गया वो
ये भी
तो
ग्रो करेगा, ये भी ग्रो
सेंसिटिव।
करेगा। ठीक है, तो इसको so these are so the bacteria which will have these type of so the bacteria which will have these type of plasmid, your these recombinant plasmid, they are called recombinants. ठीक है, सिर्फ इसी को recombinants बोलेंगे। और ट्रांसफॉर्मेंट्स मतलब ये सब ट्रांसफॉर्मेंट्स है। जो ये इस तरीके का प्लाज्मिड लिया है वो भी ट्रांसफॉर्मेंट है। The bacteria which have taken self-ligated plasmid, they are also transformants, and the bacteria which have taken up the plasmids which are carrying the wrong gene of interest, they are also transformants. But only these are called the recombinants. So you have to screen for. So once you get the colonies, that does not mean that all your all these bacteria will have your gene of interest, or they will have the recombinant plasmid in the recombinant plasmid will be present in the bacteria. So I have to screen that which are the my recombinants and which are the non-recombinants. So you will then use the particularly select the recombinant plus bacteria and use it for further experiments. Is it clear? Transformants मतलब ये सभी हो गए, जिसने भी तुम्हारा सेल्फ लाइगेटेड वेक्टर लिया वो भी ट्रांसफॉर्मेंट्स है, जो गलत प्लाज्मिड ले लिया वो भी ट्रांसफॉर्मेंट्स है। लेकिन रिकॉम्बिनेंट मतलब जिसने सिर्फ रिकॉम्बिनेंट प्लाज्मिड लिया, दिस इज कॉल्ड द रिकॉम्बिनेंट प्लाज्मिड
Sir,
having your gene
means
of interest.
recombinants are one type of transformants.
Yeah, they are one type of transformants. So इसीलिए there are two different slides here. So this slide is for differentiating between the E. coli cells which have your plasmid and which do not have your plasmid. Okay, but recombinant screening. Recombinant screening means you are particularly screening for these type of bacteria. ठीक है, तुम्हें सिर्फ ये वाले बैक्टीरिया चाहिए, ये नहीं चाहिए, ये नहीं चाहिए। ठीक है, they are one type of transformants. Yes, you can say that. Okay, you have to screen for this. So it is necessary to determine which of the transformed colonies comprise cells that contain recombinant DNA molecules and which contain self-ligated vector molecules. Okay. With most cloning vectors, the insertion of DNA fragment into the plasmid destroys the integrity of one of the genes present on the molecule. So you must have studied about insertional inactivation in class 12. Have you heard about of this term?
Yes, sir.
So what is the meaning of this term, insertional inactivation?
Means when we are inserting the desired gene, then the means all further processes will happen. So after inserting that, means it was an example of galactosidase, I think.
Beta galactosidase. Okay. So
Blue
insertional
white staining.
inactivation. Yes, you are partly correct. So insertional inactivation actually means that, so for example, you have a plasmid. Okay. This plasmid has a gene, has a beta gal gene. Okay. This plasmid has a beta gal gene. So this beta gal or lac-I gene. So lac-I gene basically codes for this beta galactosidase enzyme. So now you have done such a cloning that your gene of interest has now you have cloned your gene of interest inside this beta galactosidase gene. तो बीटा गैलेक्टोसाइडेस का जीन के बीच में तुमने अपना जीन ऑफ इंटरेस्ट क्लोन कर दिया. So now can the functional beta galactosidase protein be produced? तुम अगर किसी जो जीन के बीच में अगर तुम
नहीं
कोई जी
नहीं नो सर।
नहीं होगा ना वो एक्सप्रेस, तो ये इसी को बोलते हैं इंसर्शनल इनएक्टिवेशन. सो इफ यू क्लोन योर जीन ऑफ इंटरेस्ट इन अनदर जीन, सो दैट इज व्हाट इज रिटर्न हियर. द इंसर्शन ऑफ अ डीएनए फ्रैगमेंट इनटू द प्लाज्मिड destroys the integrity of one of the genes present on the molecule. Okay, so recombinants can therefore be ligated. So explain in this slide. So for example, this is your plasmid. Okay, this is your plasmid, and this plasmid has a beta gal, has a gene which codes for beta galactosidase. Okay, this plasmid has a gene which codes for beta galactosidase. Now you have performed a cloning in such a way. So for example, this also has one equar1 site. This beta galactosidase, for example, consider that beta galactosidase gene has an equar1 site. And then when you treat this bacteria, this plasmid with equar1 enzyme, what will happen? This plasmid will be cleaved. Okay. Now in this equar1 site, you clone your gene of interest. You have cloned your gene of interest. There will be some portion of the beta galactosidase gene also. Okay. So now what will happen? So this was this plasmid when it was this plasmid when it was transformed in a bacteria, the bacteria was able to express the functional enzyme. But once you have cloned your gene of interest inside this beta galactosidase gene, then this bacteria can no longer express the beta galactosidase protein or beta galactosidase enzyme. So this is called insertional inactivation, and you can use this method for screening recombinants and non-recombinants. Okay, is this point clear?
How sir? How
So
is
how is it possible?
means
Okay,
means sir? I am
okay,
explaining.
so I'll
So means
explain you through this figure. You see this figure. Okay, so do not see all this, whatever it is written, you do not have to read. You see these two figures. Okay, so this is a PBR322 vector. Okay, this is a PBR322 vector. Now, this PBR322 vector has two antibiotic resistant genes. So in green, this green, marked in green, this is your tetracycline resistant gene. And the red, the part which is marked in red, this is your ampicillin resistant gene. Okay, now what you have done now in this tetracycline resistant gene, there is a BAMH1 site. Okay, now what you have done is you have cloned your gene of interest in this BAMH1 site. Okay, so see this figure, the second figure now. So you have cloned your gene of interest in this BAMH1 site. Now what will happen if you transform? So if you transform a bacteria, so now you have transformed the bacteria with this vector. So this is your vector, plasmid vector, and you have transformed this in a bacteria. And you have transformed this vector in another bacteria. Okay, so this is your bacteria. You have transformed this plasmid vector into this bacteria, and you have transformed this type of plasmid bacteria vector in this bacteria. Now you tell me that uh, this type of bacteria, yeah, can it grow in ampicillin or can it grow in tetracycline? So what happens? So if you see this, समझ
ampicillin.
आ रहा है? हाँ,
Yes sir.
तो हाँ, तो जैसे ये तुम अगर ट्रांसफॉर्म कर दिया ना, तो ये बैक्टीरिया अपना टेट्रासाइक्लिन रेजिस्टेंस का प्रॉपर्टी खो देता है. ठीक है, क्योंकि इसमें जो कुछ भी एंजाइम बना रहा होगा ना, टेट्रासाइक्लिन रेजिस्टेंस क्यों आ रहा है? क्योंकि ये कुछ प्रोटीन बना रहा है जो ये टेट्रासाइक्लिन का इफेक्ट को ये कर रहा है, इनिबिट कर रहा है. ठीक है, या फिर कोई एफ्लक्स प्रोटीन बना रहा है, या जो भी एंटी माइक्रोबियल रेजिस्टेंस का जो भी मैकेनिज्म्स होता है. So that is helping it to have resistance against the tetracycline antibiotic. Okay, so once you have cloned your gene of interest in this site, this bacteria will lose the tetracycline resistant property. Okay, so these type of bacteria, the recombinant, the bacteria which are having these recombinant plasmids, they can grow only in ampicillin supplemented media. They will not be able. So if you prepare an agar plate having both ampicillin and tetracycline, then only the bacteria which are having these type of recombinant plasmid, only they will grow. Okay. Sorry. So if you are growing it in a agar plate having ampicillin and tetracycline, then only these type of bacteria will grow. Only these type of bacteria will grow. These will not grow because they are sensitive. They have now become sensitive to tetracycline. Is it clear?
Yes, sir.
ठीक है. तो ये जो बैक्टीरिया होगा ये सिर्फ तुम्हारे ये this can only grow in ampicillin supplemented media. They cannot grow in media having both ampicillin and tetracycline. While these type of bacteria, they can grow both in ampicillin and tetracycline. Okay, so in this way, you can screen between recombinants and non-recombinants. So the recombinant bacteria are those bacteria which are growing in only in ampicillin. Okay,
Sir,
but not in tetracycline.
why this is happening, that when we are putting the petri plates in ampicillin plus tetracycline medium, then though it is ampicillin resistance, then also it will not grow.
So, so if
मतलब सर
you
टेट्रासाइक्लिन सेंसिटिव है पर एम्पिसिलिन रेजिस्टेंट तो है ना, तो थोड़ा मोड़ा तो ग्रो करना चाहिए सर.
नहीं नहीं ऐसे नहीं होता. ये जो अगर टेट्रासाइक्लिन नहीं ऐसे थोड़ा मोड़ा नहीं ग्रो करेगा. ये अगर एम्पिसिलिन अगर टेट्रासाइक्लिन सेंसिटिव है तो अगर टेट्रासाइक्लिन ऐड कर दोगे तो नहीं ग्रो कर पाएगा. ठीक है.
ओके सर.
ओके. सो if it is sensitive to any one of the antibiotic, then it cannot grow. It so this is what is explained here. So BAM each one, for example, cuts PBR 322 at just one position within the cluster of genes that code for resistance to tetracycline. So this BAM each one site is present in such a location. It is present in this tetracycline resistant gene. Okay, it is present in the tetracycline resistant gene. So what happens? So as a recombinant PBR 322 molecule. So this is a recombinant PBR 322 molecule. So this recombinant PBR 322 molecule, it will carry an extra piece of DNA in the BAM H1 site. So in place of this BAM H1 site, you have cloned an extra piece of DNA. So now this once you have cloned this gene of interest in this tetracycline resistant gene, then this bacteria is no longer able to confer tetracycline resistance on its host, as one of the necessary gene is now disrupted by the inserted DNA. Okay, so the cells containing this recombinant PBR322, they are still resistant to ampicillin, but are sensitive to tetracycline. Okay, so colonies. So the colonies that grow in tetracycline supplemented media consist of cells that carry the normal PBR322 with no inserted DNA and therefore a functional tetracycline resistant gene. So the point of difference between these bacteria and this bacteria, or bacteria having this vector and bacteria having this vector, is that one of the bacteria will be sensitive to tetracycline and the other will be resistant to tetracycline. So if you grow, if one of the if the bacteria grows in tetracycline, it means that this bacteria are non-recombinant. It does not have your gene of interest. But if the bacteria is not able to grow in tetracycline, it means that my bacteria is tetracycline sensitive and it has the gene of interest. Okay. So that the colonies that do not grow on tetracycline agar are recombinants. Okay. So in this way, you can use the antibiotic resistant gene as a for selecting between recombinant bacteria and non-recombinant bacteria. So now answer this question. So we read lot of theory. Now try to answer this question. So do you remember the plasmid isolation procedure which we discussed? So how do you isolate? So so if you see a bacteria, so inside the bacteria will have its own genome. Okay. It will have its own genome. It will have a lot of other proteins and lipids in its cell, and it will have a plasmid. It will also have a plasmid. So how will you isolate the plasmid from all these different components which are present in a bacteria? So how will you? So this is called plasmid purification. So there are kits available, commercial kits by which you can isolate the plasmid. There was a demo also which was shown in the lectures. In week one, it was shown that how will you purify the plasmid from a bacterial cell. So for this, what you have to do? First, you have to lyse the cell. Okay. So once you lyse the cell, so for this, what is used? Lysis buffer. Okay. So for this, what is used? A lysis buffer will be used. So this lysis buffer will have SDS. Okay, so let's discuss this plasmid isolation a little bit. Okay, so there are for plasmid isolation you require three different buffers. Okay, first is solution one. So this solution one, what does it have? This solution one will have, it will have 10 millimolar tris. Okay, 50 millimolar glucose and EDTA. EDTA is around zero point 0.5 millimolar EDTA. So this is called the desuspension buffer. Okay, this is called the desuspension buffer. So the bacterial pellet that you have obtained, this bacterial pellet, so you grow a culture. Okay, 10 ml culture is a 10 ml culture of bacteria is grown. Then this 10 ml culture is centrifuged. It is centrifuged and you get a bacterial pellet. Now first what is done? Well, this bacterial pellet is first resuspended in solution one. Okay. Now now in the next step, what is done? In the next step, you add the lysis buffer. Okay, so this lysis buffer has two components. One, it has NaOH. So it has zero point 0.2 normal NaOH and it has SDS, 1% SDS. So what this NaOH and SDS does? So SDS, what it will do? It will rupture the bacterial membrane. Okay, it will rupture the bacterial membrane, and so once this bacterial cell wall is ruptured, all the components of the which are present inside the cell, they will come out. Okay, so all these components, now these components will have different enzymes. It will have RNAs, DNAs. So these RNAs, DNAs, these enzymes have different metal centers. So when you add EDTA, to this solution, to this culture. This EDTA, what it does, it chelates out. It chelates out these metal centers and it inactivates these enzymes, RNAs, DNAs, all this nuclease activity is restricted. And this NaOH, what does this NaOH do? This NaOH denatures the DNA. So this NaOH breaks the hydrogen bonds which are present between the polynucleotide strands. So this NaOH, what will it do? It will denature both the plasmid DNA and it will denature the, so it denatures both plasmid DNA and the chromosomal DNA. So it denatures both the plasmid DNA and chromosomal DNA. Now in step number 3, you add neutralization buffer. Okay, so in the neutralization buffer, what is present? What are the components? There is potassium acetate and there is acetic acid. Acetic acid. So neutralization buffer, why? Because you had added a base in the previous buffer. Now you are adding an acid. So this high pH, at high pH, this hydrogen bonds get broken. So once you add this acetic acid, this it the DNA. What happens is the plasmid DNA. The plasmid DNA, because it is very small compared to the chromosomal DNA, this plasmid DNA can renature. This plasmid DNA gets renatured. Okay. And also it is supercoiled, and because of all the supercoiling present in plasmid DNA, it can easily renature. But the chromosomal DNA cannot renature in presence of the neutralization buffer. And this potassium acetate, what it does, it precipitates the chromosomal DNA and other proteins which are present in the lysate. Okay, while this plasmid renatures, and now you what you do is you take a column which has affinity for this plasmid, and then you pass the solution through this column, and then elute the DNA, the plasmid DNA, while these chromosomal DNA and all these get precipitates, and these precipitates are removed. So basically, this EDTA, TCDTA buffer is used in resuspension to inhibit the nuclease activity. Okay. So when you lyse the cell, all these different RNAs, DNAs, these are also, this will also be present in your lysate, and they can denature, or they can degrade the plasmid DNA. So to inhibit the nucleus activity, this EDTA is this EDTA is present in this solution one. Okay.
So, but EDTA we are adding in solution one to enhance the cell lysis first, and
Uh, yeah. Yes. Tell me, EDTA you are adding for enhancing the cell
for
lysis.
yes, sir, because a solution after solution one, in the solution two, the plasmid will get denatured. So why
Plasmid will get
we are
denatured.
yes, after adding solution two.
So everything is taking place in a test tube, right? So you have the bacterial cells. You first add solution one. You first add solution one. This solution one will have EDTA. Okay. And then you resuspend the cells. You resuspend the cells. Then after adding the solution one, you add solution two. So when you add, once you add the solution two, your cell gets ruptured. Your cell wall gets ruptured, and all the contents which are present inside the cell, they come out. So now this EDTA, which was present in solution one, that will inhibit the nucleus activity, right? That will inhibit the all the DNAs and RNAs activity, which are present inside the cell. Is it clear now?
So, but
So we are
after
not
doing, mhm.
after what?
After
So this EDTA is
taking,
already present now. EDTA is already
so we
present,
will
and then
not
you are
remove,
lysing the cell.
we will not remove the solution one
No, no,
in the
we do not
solution
remove the
one.
solution one. No, we
Okay,
do not remove. That is why it is called resuspension buffer. So when you centrifuge the culture, you will get a bacterial pellet. Okay. Now to resuspend the bacterial pellet, you add the solution one. So so you get a now you again you get a bacterial sorry resuspended bacterial culture. Okay. And now you add the solution two. You do not discard solution one. Okay. Now after adding solution two, you have to again spin it for 10 minutes. So after spinning it for 10 minutes, what happens? These chromosomal DNA and all the protein and lipid fragments, all of these get precipitated. And your plasmid DNA remains in the supernatant. So you take out the supernatant, and then okay, take out the supernatant, and then you pour it in the column for elution.
sir. What is the need of inhibiting the nucleus activity?
So if you do not inhibit the nucleus activity, then what will happen? Your plasmid might also get degraded, because when the cell is getting ruptured, all the RNAs, DNAs which are present in the cell, they will come, they will also be present in your lysate along with your plasmid. So these
But after
different
that, we are
enzymes
adding
into
neutralization buffer. After
neutralization.
neutralization,
Yes, this neutralization buffer is not for neutralizing EDT or anything. Neutralization buffer we are adding so that, so because when you are adding NaOH, both your plasmid DNA and chromosomal DNA, both these DNA are getting denatured. So both these DNA will converted from double-stranded DNA, they will get converted into single-stranded DNA once you add NaOH.
no, sir. My
Okay.
question is that what is the need of inhibiting nucleus activity? At last, we have to means in the while we are adding the second buffer, at that time, the plasmid and DNA both are getting denatured. So,
Yeah,
so,
both
so
are
why we are
getting, they are
why we are
getting,
adding
they are getting denatured, but they are not getting degraded. Denatured means they are the DNA is getting converted from double-stranded to single-stranded. But if you do not inhibit the nucleus activity, then what will happen? This DNA will get thus after you get this single-stranded DNA, then this single-stranded DNA will be converted into short DNA fragments. It will get chopped
okay,
off.
okay,
It
got
will
it, got
get chopped off, and yeah, it will get DNA fragments. So then your entire plasmid will get degraded. The- You will get DNTPs in your solution then. So if it gets chopped up, you will only get DNTPs, DATP, DDTP, DTP, only these things you will get. Okay, but you want your intact plasmid. That is why you inhibit the nucleus activity. The plasmid does not get degraded. Okay, so this is also one of the techniques which is done routinely in any lab. Okay, so these things, if you join any lab, you can do this, perform this experiment. Then, so this is option number C. Then common use of ethanol during plasmid isolation is. So you have read in class 12, no? You have
precipitation.
read in class 12, yeah, precipitation,
Okay.
chilled ethanol. Yeah, so this is very common question. That yeah, ethanol is used for 100% ethanol if you add 100% ethanol or zero point 0.7 volumes of isopropyl alcohol. If you guys add isopropyl alcohol also, then also the plasmid gets precipitate, and 100% ethanol has to be added 3 times the volume of the DNA of the solution. Okay. It is used for plasmid precipitation. Okay. Now RNase each is unique among other RNases. So this question has been repeated again. RNase each, when I was discussing reverse transcriptase and how do you generate cDNA, then this question
Means
was there. RNase each.
RNA, DNA, RNA hybrids.
Yes, DNA, RNA hybrid specifically. Yes, yes. Option C. Okay. So you remember how we generate cDNA from
Yes,
RNA?
yes, sir.
Okay, sir. Tell me then how is it generated? How can you
So
generate
first,
cDNA? Hmm.
we will take the mRNA and then we will
Yes, you
generate,
get the
means we will
mRNA.
make it complementary of that sequence.
Yes. How do you do that? So yeah, you will have the mRNA having this
Poly,
poly A tail, and then you add a primer. You add a oligo DT primer. No, you add a oligo DT primer, and then you add the reverse transcriptase enzyme. So this reverse transcriptase enzyme, what it does, it will extend this strand, or it will synthesize a strand complementary to the mRNA strand.
and
Okay,
then we
so
will
you
put the
have this.
RNA and then it will break
Then you
up.
will put the RNA. Yes. Okay. Okay, good. You remember this. So we will add RNA. So RNAs will create short fragments, and then you add polymerase. So generate a cDNA. Yes. So this is there. It was there in your slides as well. So this is your mRNA. This mRNA is having this poly A tail, and then you add a cDNA, not a cDNA. So this is an oligo DT primer. So this TTT primer, and then you have added a few sequences complementary to your RNA, and then you add the reverse transcriptase and 4 DNTPs. So once you add this reverse transcriptase, it is a DNA dependent RNA polymerase. So it will synthesize the complementary DNA sequence. And when you add RNase H, so RNase H, what has it has done? It has created fragments of RNA. Okay, let's create fragments of RNA. Now, using these fragments of RNA, the DNA polymerase can synthesize the complementary DNA sequences. So here, then DNA polymerase is used to perform DNA synthesis using multiple fragments of RNA as primer to synthesis new DNA strand. This method produces blunt-ended duplex DNA product. Okay. Answer to this question, it is preferred to use a bacteria of dash for making competent cells for transformation. So you know a bacterial growth curve.
Yes, sir.
So a
It
bacteria,
is increasing bacteria. Yeah, stationary phase. Sorry, log phase.
yes, log phase. So these are very simple questions. Okay. So log phase, why will you use log phase bacteria in log phase for making competent cells?
Because at that time the bacteria will grow.
Yes, the bacteria will grow, and in the log phase, what happens? The bacteria is actively synthesizing different proteins and different repair proteins. DNA repair proteins are also synthesized during the log phase. Okay, so what happens is when you are giving this heat shock and or you are giving this stress to bacteria, then then uh, to survive these stress conditions, there has to be some protein or so that these breaks, double-stranded breaks, or these repair pathways can be activated, and the bacteria can take up the DNA, right? So if you take bacteria from the log phase, then it can handle such type of stresses basically. Okay. The growth stage of bacteria has a significant impact for its ability to take up foreign DNA. The bacteria at the log phase is more active and efficient to perform DNA damage and repair than stationary phase. Okay, so all these DNA, so at these stress conditions, there will be some DNA damage. There will be some uh, damage even in the bacteria. To repair or to enhance this repairing, the bacteria from the log phase is usually taken for making competent cells for transformation. Okay. And also when you do transformation, what is done is after this heat shock, after you give the heat shock, then the bacteria is again you add a recovery media to the bacteria. So you add the LB media to bacteria. So and then you keep it for around 45 to 1 hour, 45 minutes. If there has been any any enzymes which are required for antibiotic resistance, all these gets synthesized during this time. Okay, and any recovery or any if the cells have lysed or if there are any pores in the bacteria, so all this gets recovered in this recovery phase. Okay, so after the heat shock period, there is a recovery phase that is given, and after that the bacteria is then plated on agar plates. Okay, so these are actually difficult to explain here. So once you do your experiments on hand, if you have any on hand training, then you can understand. Okay. So for now, just understand that. So in log phase, they're actively dividing, so it is used. So the bacteria cells like E. coli are treated with answer this question. So I explained just now. So for bacterial transformation, what type of salts are required? So they are treated with dash and then suddenly exposed to high temperature. This is known as the heat
Calcium,
shock treatment.
calcium
Sorry.
chloride, calcium
Yes,
chloride.
calcium chloride. Yes, good. Calcium chloride is used. for bacterial cells, for E. coli cells specifically. Okay. So yeah, this is a figure that was there in the lecture slides. So you see here, so you take a ice bucket. So these are your competent cells. Then these ampicillin resistant plasmids are added to your competent cells, and then you give it a heat shock of 42 degrees Celsius. And after this brief heat shock for 90 seconds, you quickly place it in ice again. So after this heat shock, so there is a figure missing. So you again place it in ice. Again, you place it on ice for 5 minutes. And after that, you keep this bacteria in a recovery media. So you add around, so if it is 100 microliter competent cell, then you add around 900 microliter of LB media, and you incubate at 37 degrees Celsius. Okay. 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 So the antagonist, to answer this question, the antagonist gene of lytic cycle in lambda phage. So this lambda phage is also a vector, can also be used as a vector for gene cloning. Okay, so like the plasmid vectors, so depending, actually it depends on your gene insert. So if the gene insert is small, if it is around 2.3 kb by 4 kb, so less than 3 kb, then you can clone your gene in a plasmid vector. But if it is a if your gene of interest is of 5 kb or more than 5 kb, then you have to then it is difficult to clone your gene of interest in a plasmid vector. You have to go for a lambda phage vectors. Okay, so have you read about this lambda? So this was explained actually in the lecture. So I didn't add any slide for this. So in lambda phage, there are two types of cycle. One is this lytic cycle and another is lysogenic cycle. In lytic cycle, what happens? The once the phage DNA enters inside the bacteria, once the phage DNA enters inside the bacteria, then what it this DNA from this DNA, different components of the bacteriophage are synthesized. The capsid proteins, then the tail fibers, all these are synthesized, and then and then these are assembled. These are assembled, and the assembled viral particles lyse the bacterial cell and are released into the in the media. So this is lytic cycle. While lysogenic cycle, what happens? The phage DNA after entering into the bacteria, this phage DNA integrate into the bacterial chromosome. Okay, so when the bacterial chromosome will replicate, the phage DNA will also replicate along with it. So that is lysogenic cycle. So the antagonist, so there is one repressor protein actually. So this is C1 protein. So this C1 protein is an antagonist of lytic cycle. C1 protein, it inhibits the lytic cycle while it promotes the lysogenic cycle. Okay. So this you see here. So this is a lambda phage vector. So this is again a screening strategy. Okay, this is again a screening strategy. So when this so this is C1 repressor protein. So this is the gene which codes for the C1 repressor protein. So what this C1 repressor protein does, it promotes the lysogenic cycle. Okay, so lysogenic cycle, what will happen in lysogenic cycle, you will not get any plaques because the bacteria is not disrupting the, or a viral cell are not lysing the cells. So you will not see this viral plaques, you will not observe if the particular bacteria is following lysogenic cycle. Now, if you clone your gene of interest in this C1, in this gene of C1 repressor protein, okay, then what will happen? This C1 repressor protein will not be expressed. Okay, C1 repressor protein, functional C1 repressor protein will not be expressed. Now this lytic cycle will be, this lytic cycle will be active. Okay, so you will get plaques. So you remember that this is the protein. So during an infection cycle, virus undergoes a lytic and lysogenic stages. So in the lytic phase, what happens? It is responsible for the lysis of host to release the virus particles. Okay, so the host cells are lysed and the viral particles are released from the host cell. But in lysogenic stage, what happens? It allows the replication of virus without the lysis of host. So your gene of interest, it gets integrated in the bacterial chromosome. So along with your bacterial replication, your the bacteria, the virus gene also gets replicated. So the C1 gene encodes for C1 repressor and is responsible for formation of lysogens. Okay, formation of lysogens means lysogens are bacteria which are showing the lysogenic cycle. So in the presence of the functional C1, the plaques contain unlysed host cells. Okay, so in the presence of functional C1, what will happen? The bacteria will be growing. Okay, the bacteria will be able to grow. So you will see that it will have a turbid. You can see turbidity in the culture. While if the bacterial cells are not able to grow, then the culture will appear transparent. Okay, because the virus, what it is doing, it is lysing the bacteria. So once the bacterial cells are lysed, then you will have a clear solution. There will be no turbidity, and this can again be used to screen the clone to detect functional C1 protein, or on the basis of whether this C1 is functional or whether it is absent. You can that can also be used. So you clone that your gene of interest in a phage vector, then you transform the bacteria, and then there will be homologous recombination between the your phage DNA and your bacterial chromosome, and the phage DNA will be integrated inside the bacterial chromosome, and then it can be expressed also. Okay. So then there are various other methods by which you can transform the DNA in different types of host cells. Okay, so you have read about all these techniques. Okay, so one is transfer by this calcium chloride method. So this calcium chloride or rubidium chloride, these methods are used for bacterial cell. Well, there are different other methods as well. So you can use liposome. Okay, so cultured animal cells which lack cell walls are easily transformed, especially if the DNA is precipitated onto the cell surface with calcium phosphate or enclosed in liposome that fuse with the cell membrane. So you can transfer the DNA in cells which are lacking cell wall. So in mammalian cells, if you want to transfer the DNA, then what you can use, you can use liposomes. So what are liposomes? These are lipid vesicles. Basically, they are lipid vesicles, or they are double layered lipid vesicles, where you can insert your DNA, and then these liposomes they fuse with the membrane, uh, cell membrane, and the DNA enters into the cell. Then there can be electroporation. So in electroporation, and short electric pulses are applied. And these electric pulses, what they do, they induce transient formation of pores in the cell membrane. Once these pores are created, the DNA molecules can enter the cell. Then there are microinjection techniques. So in microinjections, you see this is an image of microinjection. So there is a fine pipette, and it is used to inject DNA molecules directly into the nucleus of cells to be transformed. Okay, so this technique was initially applied to animal cells, but has subsequently been used in plant cells as well. Then, by bombarding the cells with high velocity microprojectiles. So these are your target cells, and then you bombard it with microprojectiles. So what does this microprojectiles have, or what do they contain? They contain the they are actually gold or tungsten particles that have been coated with DNA. So this unusual technique is called ballistics and has been used in several different types of cells. These are the different ways by which you can transform any host cell, or you can introduce DNA in host cell. So transformation is used for bacterial cell, but uh, transformation is not basically used for other type. If you are transferring or introducing on DNA in any other type of process, then it is not called transformation, but it is so frequently used. Sometimes people use it interchangeably. Okay, so now dash is a popular physical method of transfection that uses an electric pulse to create temporary pores in cell membrane. What will be the answer of this question? So just now we discussed, right? So it is electroporation. So in electroporation, short electric pulses are given, and these electric pulses create pores in the membrane or in the cell wall so that the DNA can be uptaken. Okay. So it is a plasma membrane is composed of lipids and proteins, and then these macromolecules, uh, these lipids and proteins, they have partial conductance to the cell membrane. So when you apply this high electric pulse, this charge, the charge which is present across the membrane, that gets disturbed. As a result, what happens? Pores are formed, and this allows easy passage of macromolecule, especially charged molecules like DNA into the cell. Okay. So these are charged molecules, DNA. So hydrophilic molecules cannot, or you have to apply some physical methods by which we can cross this hydrophobic lipid bilayer which is present, which composes the cell membrane. So in blue-white screening process, a colorful substrate is processed into a colored compound. The colored compound XGAL used in the screening method is a substrate for enzyme. So I was just now, we were telling about beta galactosidase. So this beta galactosidase, it is an enzyme. This and the substrate for this enzyme is XGAL. It can convert XGAL into a product. So there is a particular name of this product, and this product is, so when this XGAL is converted into product, you get a color. See blue colonies. You see blue colonies. Okay, because of the formation of this colored product. But now this can also be used in for screening recombinant and non-recombinant clones. So you clone your gene of interest in the beta galactosidase gene. Okay, so if you clone your gene of interest in the beta galactosidase gene, then what will happen? This beta galactosidase protein cannot be synthesized anymore. So the blue colored. So now you will have two types of colonies in your plate. You will have some white colonies and you will have some blue colonies. So the blue colonies will indicate that they are the non-recombinant bacteria. The blue colonies will mean that they are blue colonies indicate that they are non-recombinant. Non-recombinant. So these are called the non-recombinant E. coli cells. E. coli. And the white colonies will be E. coli cells having the recombinant plasmid. Okay. So these are the recombinant E. coli cells. Okay. This is also a screening method. So these are all following the principle of this insertional inactivation. Okay. So all of these are similar. So this, so the use of chromogenic substrate to detect a particular enzymatic activity is the basis to screen the desired clone. So the most popular system to exploit this feature is the blue-white screening. So in blue-white screening, what is done? A colorless substrate is processed to a colored compound. So the colorless compound is XGAL, or this XGAL is the commercial name, or it is a common name. While this what is this is a compound actually. So this is 5-bromo-4-chloro-3-indole beta-D galactoside. So this is used in the screening method, and it is a substrate for beta galactosidase. So this beta galactosidase enzyme will act on the XGAL, and what happens? It produces the colored product. Now there is another method. So so the another again another type of modification has been done. So what is done is the this beta galactosidase. This beta galactosidase has an initial N-terminal region. So around 11 to 41, 11th residue to 41th residue of the protein is important for the activity of the protein. Now in this system, the host contains the laxi gene without the initial region, whereas the vector contains the alpha peptide. So the host contains the laxi gene, which means that the bacterial chromosome will have the laxi gene. Okay, and the vector that you are using, that will have the alpha peptide gene. So this alpha peptide is basically this N-terminal region, which is required for activity of the beta galactosidase. So if a vector containing alpha peptide is transferred into the host containing the remaining laxi, then the two fragments will reconstitute to form active enzyme. Okay. And in addition, the alpha peptide region in the vector contains multiple cloning sites, and as a result of insertion of gene fragment, consequently, alpha peptide will not be synthesized to give fully active beta galactoside. That's why what will happen? The enzyme beta galactosidase oxidizes X-gal to form 5 bromo 4 indoxyl and galactoside. The indoxyl derivative is colored colonies indicate the presence of an active enzyme. So what has been done? This beta galactosidase gene is there. Now this beta galactosidase gene has an N-terminal region. N-terminal region means the a protein will have an N-terminal and a C-terminal region. C-terminal is the starting region of the protein. N-terminal is the end region of the protein. So this N-terminal region from residue 11 to 41. So this is called the alpha peptide, and this is required for activity of the beta galactoside. Now this alpha peptide region of the protein, that has been cloned in a vector. Okay, that has been cloned in a vector. Now your vector has this alpha peptide, and your bacteria has this beta galactosidase gene without this N-terminal region. So what will happen when so if you are cloning your gene of interest in this alpha peptide, then this alpha peptide will get inactivated. The functional alpha peptide will no longer be able to produce, and will not be produced, and so this alpha peptide can not activate the beta galactosidase, and so you will get white colored colonies. Okay, so you see over here. So this is the figure. So you see laxi gene. So from this laxi gene produces the beta galactosidase. Okay, this is producing your enzyme beta galactosidase, and from here this 11 to 41 region of this gene has been deleted. So this is the gene. This is not the amino acid residue. This 11 to 41 region has been deleted, and it has been cloned separately. This 11 to 41 region, this has been cloned separately in your vector. This is dash, laxi dash. So this is your alpha peptide region, which has been cloned separately in your vector. Okay. Now this laxi will produce a laxi fragment, and together this laxi fragment and the beta galactosidase, inactive galactosidase will give the active beta galactosidase. And now when this active beta galactosidase is present, it will act on X-gal and the blue colored product will be formed. Okay, but if you clone your gene of interest in this laxi region, then this laxi fragment will not be produced and your beta galactosidase will remain inactive. You will get white colony. So DASH is an approach to screening where a foreign DNA is cloned within the coding region responsible for a particular phenotype. As a result of it, the gene is unable to express phenotypically. So what will be the answer? So it is an approach to screening. So all these techniques which we have been discussing, they are all following this approach only. So is the chlorine DNA is being cloned within the coding gene responsible for a particular phenotype. So like this beta galactosidase that was responsible for a particular phenotype. And what was the phenotype? The phenotype was that you were getting blue colored colonies. That is the phenotype that you were getting. Now you have cloned your gene of interest within this coding region. So what has happened? And this gene will get inactive. or this lag Z dash that we were discussing that just now, that will get inactive. So the gene is unable to express now. So that is called insertional inactivation. Okay. Insertional inactivation. Insertion of something is resulting in inactivation of the product, or that product is not being formed. So that is what insertional inactivation. So insertion of the gene is resulting in inactivation of the activity of the enzyme. So that is why this is the term. Okay. So for in insertional activation, what is done? It is an approach. So these are nowadays for screening recombinant versus non-recombinant. You directly isolate the plasmid, you send it for sequencing, and you will get that whether your plasmid has your recombinant or has your gene of interest or whether it does not have. So now sequencing is very routinely used and it is not expensive as well. So people do not go for these techniques. These were used earlier and offered. And other than that, commonly what is used is PCR method is used, and there is restriction digestion. So using restriction digestion and PCR, you can identify whether your clone or whether your plasmid has the gene of interest or whether it is non-recombinant. Okay. So this is the mechanism. This is what is insertional inactivation. So this is your laxi gene. It is giving you functional beta galactosides, and this functional beta galactosides is converting this X-gal into a product which is giving blue color. But if you insert interest inside this laxi gene, then this functional beta galactosides will no longer be produced. It will become non-functional, and so you will get colorless colonies. Now, different enzymes are used to screen transfected mammalian cells via reporter gene assay, below which is the correct pair between a gene product and its catalyzed reaction. So these are some fact-based questions that you have to remember. So this is actually a table of what are these genes and what are the gene products. So ampicillin resistant gene, it codes for this gene product, beta lactamase. And when this beta lactamase is produced, this enzyme degrades ampicillin. That is why it is resistant to ampicillin. The bacteria is resistant to ampicillin. Similarly, this kanamycin, it codes for a protein or it synthesizes the protein neomycin phosphotransferase 2 that results in covalent modification of kanamycin. And the kanamycin is now no longer active or no longer, it is now not able to inhibit the bacterial growth. Okay, because there has been some covalent modification in this kanamycin. Structure gets changed. Okay. Similarly, tetracycline, the ribosomal protection proteins. So, so there are different types of proteins that are synthesized which confer resistance to the bacteria. Similarly, for the screening of transfected mammalian cells, there are these different gene products. Okay. So this LUC codes for luciferase. And this luciferase, if it is produced, the reaction it catalyzes converts luciferin to oxyluciferin. Similarly, this PHOA it codes for alkaline phosphatase, which results in release of inorganic phosphates. So these different methods can be used for screening of transfected mammalian cells. Okay. So you see here, yeah, so these are the tables. So luciferase converts luciferin to oxyluciferin. So this is the only correct option. So if you beta galactosidase, this is wrong. So beta galactosidase, it's converting O nitrophenol beta galactoside, O nitrophenol beta, not beta glucose, beta galactoside to O nitrophenol and galactose. So that is why this is wrong. And this alkaline phosphatase, it is introduced organic phosphate in a reaction is not introduced, it is releasing. Because alkaline phosphatase will cause the release of inorganic phosphate. Okay. Now, which is the following method? Which of the following? Now, these things I had discussed earlier, so I will not be discussing it again. So, Maxam-Gilbert method and the Sanger sequencing method. Sanger sequencing. So, these two sequencing methods were covered in week 3 or week 4. So, you can go through the lectures of week 2 and 3. So they have been explained in the lectures, video lectures also. So this Maxam-Gilbert method is a chemical sequencing method. So different type of chemicals are used. So there are reagents, and you need to remember the name of these chemical reagents. Okay. So it is a Maxam-Gilbert is a chemical sequencing method. Okay. Sanger method you use there is dideoxy nucleotide triphosphates are used in Sanger method. So dideoxy means that has the 2' and 3' OH. Both are both are absent. Both are absent. So you set up 4 different reactions. In one reaction, you add DD-ATP plus the other DNTPs. In another reaction, there is DD-TTP and the other DNTPs. Then in the third and fourth, similarly, you add one DD-NTP to each of the reaction, and then you do the you catalyze this. So this polymerization reaction. So whenever this DD-NTP will be introduced, and reaction will stop. Which chemical is used for cytosine alteration? So it is hydrazine. So these things you have to remember. I cannot explain. These are here. There is nothing to explain. So these were explained very nicely by Professor Vishal Trivedi in I think in the lecture 3 or 2. You can go through the lectures. This TRP lag promoter is a hybrid promoter when dash from lag UB5 promoter is fused with dash of TRP promoter. Now these are again some things which you have to remember. Right, these are fact-based questions. Okay, so this is the inducible T7 inducible occurrence expression system. So what is done in this T7 inducible expression system is that so you have your so this is your bacterial host. This is your expression host where you want to express your protein of interest. Then this is your chromosomal DNA. Okay. Now what is done? This chromosomal DNA. This chromosomal DNA has this T7 RNA polymerase gene. Okay. So this is having this T7 RNA polymerase gene. And this T7 RNA polymerase gene is under the control of LAC-UB5 promoter. This is LAC-UB5 promoter. Okay. LAC-UB5 promoter, and then there is a LAC-I gene also, which codes for a repressor, which will go and bind to this region, and it will inhibit the transcription of this T7 RNA polymerase. And the plasmid, where you have this, you have cloned your gene of interest that has a T7 promoter. Okay, so that has the T7 promoter, and your gene of interest is in control. So is controlled by this T7 promoter. So this is your gene of interest. So this is your T7 promoter, and this is your T7 terminator. So when you do not, when IPTG is not added, this T7 RNA polymerase will not be synthesized because the repressor proteins bind to the LAC-UB promoter and prevents the synthesis of T7 RNA polymerase. Now, once you add this IPTG, this repressor is removed. This repressor protein is removed, and the T7 RNA polymerase starts synthesizing. And once this T7 RNA polymerase is synthesized, it will go and bind to its promoter region. And once it binds to its promoter, it will transcribe the gene of interest, and from the gene of from the mRNA you will get the protein. So this is how T7 inducible expression system works. Okay, there are other So this is one commonly used usable expression system that is used commonly or in any bacterial culture. What is used, I explained, but there are other methods also. So the promoter, actually the promoter. So what is the promoter actually will control the affinity. The sequence of the promoter will control its affinity for RNA polymerase. Okay. So there are different types of promoters used. Like T7 promoter has very strong affinity for RNA polymerase. That's why T7 promoter is used. So now this here in this case, this TRP lag together, it is called TAC promoter. So it is a hybrid promoter where -10 and -35 regions from LAC UV5 promoter is fused with the so the -10 from LAC UV5 promoter is fused with the -35. And that gives a plasmid PKK 223. Okay. So these are different engineered bacterial strains have been produced which are having these type of promoters in their chromosomal DNA. So there are these vectors have been designed which are having high affinity for these RNA polymerases. So this is a vector PK223. So this has so this the promoter of these of the vector, it has a tag promoter. Okay. So any so if it is any expression vector, so if any for expressing the protein in a host cell, you will clone your gene of interest in an expression vector, and this expression vector will require certain has certain requirements. It needs a promoter, it needs a ribosome binding site. So all these things have to be present so that the gene can be transcribed and the mRNA can be translated into protein. Okay. So dash and electroporation is the popular method for transformation of yeast. Now for bacteria it was calcium chloride. For yeast it is lithium acetate. Lithium acetate and electroporation has to be used together for transformation of yeast. So these things are these are some actually do not require much understanding. You have to remember these things. So what are the different chemicals which are used for transformation of yeast? What is used for transformation of bacteria? Okay. So transformation in yeast requires this lithium acetate single-stranded DNA and PEG method. So in this method, the yeast cells are incubated with a transformation mixture, and this mixture has lithium acetate, PEG 3500, and single-stranded carrier DNA. And then the foreign plasmid, you will add the foreign plasmid and incubate at 42 degrees Celsius for 40 minutes. For 40 minutes. So in case of bacteria, it was only around 90 seconds, 1.5 minutes. But here incubation time is more. Okay. So these are all for today. So, so I hope all the concepts or if you have had any doubts, I could cover most of them. Okay. So, so I will end today's session now. Thank you, everyone.